Welcome to the Ricci Flow Nutrition Podcast. My name is Cameron Borg. On this episode, I had the pleasure of speaking with Gábor Somlyai. Gábor is a molecular biologist and world-leading expert in the area of deuterium in biology. He has been working for over 30 years to research and publish his findings on the role of deuterium depletion in cancer. Gábor graduated as a biologist from the University of Szeged in 1982. He received a scholarship from the Hungarian Academy of Sciences while studying for his PhD and defended his thesis in molecular biology in 1988. In the wake of the Hungarian Nobel-prize winning scientist Albert Szent-Györgyi, who said that the true cause of cancer should be looked for at the submolecular level, Gábor began his examining the biological importance of naturally occurring deuterium as a senior research fellow at the Hungarian Institute of Oncology in 1990. In 1993, Gábor established HYD LLC to carry out cancer research and drug development based on the proprietary procedure called deuterium depletion. He has two books, Defending Cancer, published in the year 2000, and Deuterium Depletion, A New Way in Curing Cancer and Preserving Health. Gábor is a holder of numerous international patents and author of over 40 scientific publications and is a highly sought-after speaker at international conferences. I feel very honoured to have had the chance to speak with Gábor, as I feel like his discoveries and work lay the foundation for understanding a major driver of diseases including, but not limited to, cancer. For the sake of this conversation, I feel it necessary to quickly outline a little bit about what we'll be discussing so it's easier to follow on. So, all elements on the periodic table have different isotopes, slightly different configurations of the same atoms that have slightly different chemical characteristics. Hydrogen is the simplest atom with one proton and one electron. But hydrogen also has another stable isotope called deuterium with one proton, one electron, and one neutron. This is still a type of hydrogen, but it is double the atomic weight and interacts differently in chemical reactions. In our body, approximately 1 out of every 6,600 hydrogen atoms, also referred to as 150 ppm, parts per million, is a deuterium atom. Our cells are exquisitely tuned to this deuterium-hydrogen ratio, and they use this ratio as a foundational signal in cell division. If deuterium levels become elevated, changes occur in cell growth and massive shifts in genetic expression result. Gábor's work shows that lowering the deuterium level effectively inhibits the growth of cancer cells, as their signal for growth is essentially impeded. By using deuterium-depleted water, called DDW in this conversation, we can lower the relative abundance of deuterium to hydrogen in the body. I hope that's enough to help you enter this conversation with ease. With all that being said, I hope you enjoy this episode. So thank you so much for coming to speak with me. You started this work a really long time ago, actually. I think you started doing studies in the early 90s. When did you come to this idea that there was a submolecular regulatory system, something really foundational that was controlling the way that cells behaved, and particularly in the way that they divide and reproduce?
The word submolecular, first I hear when I was a teenager, and Albert Szent-Györgyi, who is a Hungarian Nobel laureate, mentioned in his TV interview. So he was a very clever guy, and he recognized that it's impossible that the life which is so complex, and even that complexity is so harmonized, each chemical reaction and cell cycle. And he supposed that maybe not the big protein should be able to manage that, but must be a small particle. And then he suggested that electron could be this small particle. So I kept it in my mind, and one day the idea came to my mind that maybe the positively charged hydrogen also can be that very small particle. And then I make a note, and that was in 1976, May 21st. And then I kept it in my mind that the hydrogen, and 4 years later I was a student at the university on biology faculty, and the idea came to my mind, there is heavy hydrogen. And then so you cannot explain that, how sure can be the people in a given second that maybe this hydrogen-deuterium ratio should be responsible to organize the basic mechanism in the living organism. And after that, I had to wait 10 years. I started and I continued my work on a DNA work. I couldn't find a job on a cancer field when I graduated. And then I was a scholarship holder at the Hungarian Academy of Sciences. Then I went to Germany. Then I went to United States. And when I came back, I quit my job and I started to find again a job on a cancer field. And that started in 1990. So it was 33 years ago. And we published our very first paper 30 years ago in 1993. Mm-hmm. And the title was that paper that the nature occurring deuterium is essential to keep the normal growth rate. Because in that paper, we published that the in vitro culture, the cells couldn't grow so fast when the media was prepared with deuterium depleted water. And in a mice study, 60% of the mice were healthy at the end of the experiment, although they were transplanted with human breast cancer. So that was the very first paper we mentioned, that how important can be the nature occurring deuterium in the living organism and in the cells.
When you started to first think about the role potentially of the positively charged hydrogen, the proton, regulating these types of things happening in the cells, was your first thought, did you start to think about cancer straight away? Was that the first sort of application you thought?
Yeah, because one of our neighbor died in cancer when I was a kid. And it was very sad to see the process and how finally, of course, the oncology therapy wasn't, and unfortunately still not good enough to cure head and neck cancer patients. So that initiated in my mind that the cancer is something which is very serious. And I hear my parents and the adults around me that everybody was worried about the cancer. And the reason I was listening Albert Szent-Györgyi, thinking about submolecular, because I believe that should be the way to move forward and to solve that type of problems. And I was very happy because that molecular biology started just at the 70s. So I was very young at that time, and the very first book I read, it was about the DNA and the genes. So it was how much we know today comparing with the knowledge what we possessed 40 years ago. It's amazing. So but the reason that I was thinking about to solve the cancer problem. So that was for sure.
Yeah, that's fascinating. Great reason to try and make some progress in that regard as well. I guess it makes sense that deuterium and hydrogen have been around for a lot longer than life on Earth. So it makes sense that when cells first started to grow on the planet, that they had to have something that was a very solid foundation to use as their regulatory mechanism. And the deuterium hydrogen ratio is sort of this more or less unchanging thing that can be used, that can be exploited for purposes like cell division. So it kind of makes sense that ratio is so important. So what kind of ratio are you looking at in natural environments? Like how much deuterium is there in rainwater, in seawater, and in ourselves?
It's a good question at the start. Yeah, so we can consider, we use the ppm, parts per million. If we take 1 million hydrogen in a surface water is about 150 deuterium. So out of 1 million hydrogen, there is about 150 deuterium. But that can be different depending on where we live. So for example, in Australia, which is pretty close to the equatorial area, the concentration should be about 155, because this is the deuterium concentration of the ocean. And as the clouds appear in the equator area and they are moving to the poles, the heavy water easier fall down from the clouds and the D level is decreasing. So the farther from the ocean, the higher the altitude, the lower the deconcentration of the surface water. In Europe, where we live in East Middle Europe, we have about 148 ppm deuterium concentration. But when we go to the Norway, it should be maybe lower than 140 ppm. So this is a range where the human population is living. Go to the North Pole, it should be about 100, maybe 10, 120, 125 ppm.
Yeah, right.
And the other, okay, this is a ppm, but we explain it in millimole. And this is a key message that in our body, the deconcentration about 13 millimole. And if we compare it to the calcium, which is 2.5 millimole, the magnesium is about 1.5 millimole. That can be very interesting that nobody investigated the possible role of deuterium, even its concentration is pretty relevant because the 30 millimole. And I guess the reason that nobody investigated that when they look at the numbers, 6,600 to 1, the ratio between hydrogen and deuterium. And I suppose that was the reason that people believe, let's ignore it. What can the life do with only 6,600 to 1 ratio with the deuterium? And this is the reason, I guess, it wasn't investigated. Maybe it has some role in the living organism.
So approximately for every 6,600 hydrogen atoms, there's 1 deuterium atom. So it kind of feels like, how can that really play any sort of role in cancer? But perhaps we're finding ourselves in a position now through a variety of different means, dietary being one of them, that we're consuming and putting more deuterium in the body than probably evolutionarily we would have had. Is this one of the things that you think is contributing to increasing incidence of cancer throughout the world?
Sure, yes. So I think during the evolution, the bacteria doesn't count whether is there deuterium or not, because the bacteria is so simple, short DNA, couple of thousand genes. But as more and more cells grow together and started to organize the life, after reaching a complexity, it's required a new level of the regulation. And that what the eukaryotic cells used out, that there is a heavy hydrogen, and then they finally used out that possibility. Mm-hmm. And if you're thinking about as a chemist, they cannot imagine why should be any role of deuterium if this is the ratio. But the life, for a very small changes or a very small quantity of the deuterium can give a very strong signal, because which is clear that given proteins, for example, can make difference between the hydrogen and deuterium. So can be a discrimination, and there is a discrimination in the living organism, the membrane transport, or for example, the ATPase do not accept deuterium as a substrate. And that way, that's very low concentration can give a very strong signal for the cell what to do and what not to do. And this is the reason finally the life could develop, and that beautiful life appeared on the earth, and so complex living organisms can be on the earth.
It seems like life takes deuterium pretty seriously. I've heard you speak about looking at different fats, and fats seem to actively discriminate and put hydrogen and deuterium in specific parts on the carbon chain. And it seems like they do that very, very purposefully. I'm not sure if we know why they do that, but nature does take this hydrogen deuterium problem very seriously. And obviously, fats tend to have lower amounts of deuterium when compared to proteins, and proteins less than carbohydrates as well. So our shift towards maybe more carbohydrate food compared to our ancestors is also contributing to this increasing level. Is this correct?
Yeah. And this is the question, the answer for your question, that the human society had a different diet 100 years ago and 1,000 years ago. And the big change was in the last 60, 70 years when it was said that the fat is bad. Don't eat fat because the high cholesterol, because the heart is, and so on. And it means that our deconcentration depends on what is the D level of the water we drink and what is the composition of the diet. Because everybody every day synthesizes about 0.3, 0.4 liter metabolic water. And this metabolic water, the hydrogen coming from the carbon, which can be carbohydrate, protein, and lipids. And depending on what is the diet composition, how you cover your calorie intake, that will determine the deconcentration of the water, what the mitochondria synthesize. And this is what you said, the carbohydrate will generate 150 ppm water, which is normal. The protein maybe 130, but from the fat, we will produce 180, hardly 118, 109 ppm metabolic water. And that means the mitochondria, depending on the ratio of the calorie intake, it should be about 120, 130, 140 ppm. And that will modify and reduce the deconcentration of the water what we drink. So even in Hungary, it's about 148.
Right.
If I have a ketogen type diet, my deconcentration should be about 130 ppm.
Yeah, that's so interesting. And I think it's quite easy to forget that one of, if not the most important job of the mitochondria is to produce metabolic water. And no one gets taught that at least in my education. That was never something that was spoken about with the role of the mitochondria. You spoke briefly about the ketogenic style diet there. Clearly, there's a lot of interest and research going on with very low carbohydrate diets. Do you think that most of the benefits of a ketogenic diet are coming from this deuterium effect?
I think so, yes.
Right.
Yeah. So I first, they do not know why the ketogenic diet can be good to treat cancer patients.
Yeah.
And that should be a connection between deuterium depletion and ketogenic diet and the efficacy against the cancer. That it's finally a deuterium depletion when someone starts to consume or eat deuterium fats and reduce the calorie intake with carbohydrate, increase with fats. Yeah.
So when you're looking at the cell and you're looking how cancer cells are dividing seemingly without any sort of regulation at all, what is lowering the deuterium content in the body doing to help prevent cancer cells from growing uncontrollably?
The first thing is the diet. But I never say cancer patients have a very strict ketogenic diet, because sometimes it's not sustainable for a long time. So I recommend just reduce the carbohydrate and increase the fat intake, and that way we reduce the deconcentration. But when we talk about cancer, we need much stronger reduction in the deconcentration to trigger the necrosis of the tumor cells. And everything which can help and help so one day when we reduce the D level, that require, this is a pressure on the healthy cells and the cancer cells.
Right.
And the lowering deconcentration, which is day by day reducing, require the adaptation of the cells day by day. And in that race, the healthy cells can do it much better and easier because they have a normal metabolism. But the cancer cells cannot do that, which leads to the increase the radical concentration, and hopefully that reach the level which trigger the apoptosis. So we should balance triggering the cancer cells to the increase of radicals, and we have to do it on a long term because regarding the cell cycle, not each phase of the cell cycle sensitive for the deuterium depletion.
Right.
So those cancer cells which we're going to divide a month later, but if someone consuming DW, that cells already in a resistant phase in a deuterium depleted environment. So, but we need to get the changes in the D level even a month later. So everything which help to reduce on a long term and reaching the threshold which is necessary to trigger the apoptosis is important. And that should be the food and the water. And we must combine. And on the other hand, the higher oxygen pressure should be good because that way we can increase the oxygen, the radicals, and that can help. And we have to work on, for example, do not combine deuterium depletion with antioxidant. I mean, vitamin C, E, A, selenium, because that way we can help the cancer cells catching the radicals. We will not reach the threshold to trigger the apoptosis.
Yeah, it's interesting you mentioned that about oxygen. I hadn't heard you mention that before. Does that mean that you think that combining the treatment protocols that you describe in the book with hyperbaric oxygen would be a good idea to help the oxidation and the necrosis of the tumor?
I guess so, yes. Yeah.
Okay.
Yeah.
That's so, very interesting.
We have followed thousands of cancer patients and always try to figure out why these people respond very well and why the others not.
Yeah.
And the most obvious was that taking too much antioxidant that diminished the efficacy of DW. And later, Professor Zubarev from the Karolinska Institute published a paper proving that, how important the radicals. And because the radicals is important, the oxygenation and the high pressure of oxygen can help us to get a better efficacy with DW.
So I'm going to lay out my basic understanding of what's happening when cells initiate a dividing event. So maybe they're influenced by growth hormone that binds to the membrane, and this induces the action of a sodium proton pump in the cell membrane, where the cell wants to bring in sodium and export protons. And from my understanding, from what I've read in the book, this transporter is very, very picky about whether it has deuterium or hydrogen. It really doesn't take deuterium at all. And by exporting, am I right so far?
No, you are so good that
okay.
You should continue it. You know it as good as me. Yeah, and it's clear that you are educated in that topic. Yeah.
Yeah. So the deuterium content in the cell, the ratio is increasing, and that's essentially the signal for the cell to divide.
Yeah.
Right. Okay.
Yeah. So in the very first paper, this is how we could explain the data that we could stop cell growth in vitro and save the life of the mice. That the green light for the cell when the DH ratio reach a threshold. And that can be when a growth hormone binds to the membrane, and that will stimulate the sodium-hydrogen antiport, which means the cells pick up one sodium and transport one hydrogen, and that discriminate between the hydrogen and deuterium. So it means the activation of sodium-hydrogen transport system means the increase of the DH ratio. And independently from our research and before we published our data, that was proved that the sodium hydrogen transport is activated by the growth hormone. And the other important paper was published in the Nature. Then when the ATPase gene was transported into animal cell line from the yeast, and the ATP is generating the higher pH in the cell, that cells became tumorous, caused tumor in nude mice. And they couldn't explain it. How is it possible when we know hundreds of genes having role in the development of cancer, and how is it possible that one ATPase gene makes tumorous cell? So, and the reason is that the ATPase discriminate between hydrogen and deuterium. So any hydrogen transport on the membrane inside change the DH ratio. And when the DH ratio is increasing, that change the equilibrium in each molecules. So it will spread in the cell and changing the deuterium, the hydrogen for deuterium in each molecules, in carbohydrates, lipids, amino groups, carboxyl groups, that can, for example, modify the structure of the protein, because the hydrogen bond is very important in the DNA in the RNA. So when the DH ratio is changing, that will modify the DH ratio in each organic molecules. And then they have to do everything according to the new DH ratio. And that is something like the nervous system in our body. The nervous system is everywhere. So they can collect the information, sending to the brain and going back.
Yeah.
And when we change the DH ratio, we are sending a message to all the molecules within the cell. And this is how the cell finally can organize and harmonize all the process.
Yeah, that's a really fascinating thing that I don't think you have touched on very much in the book, perhaps because it hasn't been studied very much. But as the DH ratio fluctuates, it stands to reason that all of the molecules that use hydrogen in the body, the bonds in those molecules are also changing along with it, because they may have to use deuterium, which has a much stronger bond, which is going to modulate the kinetics of the interactions of those molecules with enzymes and so forth. Does that have farther-reaching implications for all types of disease?
So we started with cancer,
Mm-hmm.
and then we had lots of data, and we saw on the patients. And one day, a patient who had cancer came to me and said his blood sugar level went down because he was a diabetic. And then we ran a rat study at the university. And that was very interesting, that first in the rat study, so we made the rats diabetic. Mm-hmm. And in the very first experiment, we used 150 ppm and 25 ppm. And one group of the rats received insulin, the other didn't receive insulin. When no insulin, it doesn't count what is the ppm of the water. But when the rats, we used insulin, then the 25 ppm reduced the blood sugar level of the diabetic rat. Right. So we were happy. And then we used other concentration, 25, 75, 105, 125, and the normal water. And then the surprising results was that not the lowest deconcentration reduced the best way the blood sugar level of the rat, but the 125 ppm, which is very close to the 150. Mm-hmm. And then we made another rat study, and then we changed with 5 ppm, 125, 30, 35, and go back to the 150 ppm. And then it was clear that the best dose for the insulin to reduce the best way the blood sugar level between 125 and 140 ppm. And if we think about, finally, this is obvious, because to change the DH ratio requires lots of energy. And we suppose that the membrane transport can change plus minus with 20, 30 ppm that level. So to reduce 150 ppm, possible to 140, 130, or maybe 125. But I do not believe that they can reduce it or change it with 50 or 100 ppm. So I think that experiment, that the best dose and the best deconcentration for the insulin to optimize the signal system is about 125, 130 ppm, what we can reach changing the diet. And this is, my grandfather deconcentration was about 130 ppm, so in the best deconcentration, which is good for the insulin. And our population eating too much carbohydrate is about 140. In our human study, phase 2 clinical trial, when we checked the D level of the blood before the experiment study, it was 146, 150 ppm.
Right.
So there was only 4 ppm differences between the 30 enrolled diabetic patients. And after giving them the 105 ppm water, they consume it for 90 days. The range was 125 ppm and 140 ppm. And the fasting glucose went down, and the insulin resistance disappeared in 30% of these people. So our life is prepared to have a D level about 125, 130, 135 ppm, not at the 150 ppm.
So it seems like we're sort of at this stage where we're starting to realize that essentially all modern diseases, from cardiovascular disease to neurodegenerative disease, at its core has mitochondrial dysfunction sort of driving the processes. So I guess, we're looking at this this stage where everything is sort of a metabolic disease. And I guess most of the research in cancer in maybe the last few decades has been probably more focused on the genetic component. And what I love about this idea about the deuterium and the hydrogen ratio is that it sort of unifies both of these ideas together and explain and makes them both right in the same context. So can you just give a little bit of background about why both the genetic and the metabolic components of cancer are right within the context of this deuterium story we're talking about?
I guess the best example, if we talk about, for example, Herceptin or Gefinitiv, the Herceptin is a big breakthrough in treating breast cancer patients, but it doesn't mean that it is good to everybody. For only those people where the epidermal growth factor receptor is multiplied. So there is a changes in the genome, which results a much higher concentration of the epidermal growth hormone receptor in the membrane. It means when a growth hormone is coming, they have much more receptor. It means they can stimulate much easily and maybe more stronger the sodium hydrogen antiport. It means for these cells easier to reach the DH ratio, which is necessary to start the cell division. So when the Herceptin is used, they cover the receptors, and that way they inhibit the activation of sodium hydrogen antiport, and that way they inhibit to increase of the DH ratio. So all the genetic changes which can impact on the DH ratio, they are a good target drugs. And these are real effective. So we see extremely good synergistic effect when someone got a breast cancer, drinking our water plus getting the Herceptin. Then extremely good response,
Mm-hmm.
fast reduction of the tumor size, and no relapse. So that should be the way. The biggest problem is that all the signal system and lots of targeted gene, this is regulated by the DH ratio. So when the cells made the decision, because the DH ratio is higher,
Mm-hmm.
then you cannot do anything with that and targeted one gene, which is in that long signal system at the end of the signal system.
Mm.
From the end, you cannot go back and stop the cell growth. So, and it's very important, the integration of deuterium depletion to the therapy. So we need to find which targeted that therapy synergize and help to intervene in the DH signal system, and which are what we have to remove because they have very strong side effect, toxic effect.
Yeah.
They maybe extend the life with extended tumors, but no any real breakthrough. So really, it's a big demand between genetic or metabolic disease. Both parties are right, and none of them is right, because the life is not organized by only the genes and by only the metabolism. Both is necessary to figure out.
Yeah,
and this is how it works.
Yeah, and I guess you make the point in the book that the DH ratio is actively shifting genetic expression, particularly when you lower the DH ratio, you're getting more beneficial genetic expression, and it's also improving the function of the mitochondria. So you're getting both the improved metabolic effects as well as the genetic working together, which makes sense why you're getting such great results, particularly when combining the deuterium depleted water therapy with conventional treatment. It seems like both of them working together are really the best outcomes that you're getting.
Thank you again for your question. Very easy and governing. Okay, so the point is that we made an experiment. It is a so-called nanostring technology, when we are able to count one by one how many copies are synthesized from a given gene. And we tested over 700 genes, more than 200 cancer-related genes, and more than 500 kinase genes. And the kinase is a target of the drug development, to finding one kinase, hopefully developing an inhibitor, and that way we can hopefully get a good cancer drug.
Yeah.
But so when we kept the cells on deuterium depleted media, normal media, and 300 ppm deuterium enriched media, and we investigated how the deconcentration modified the expression of these over 700 genes, the point was that 99.7% of the genes responded to the higher deconcentration. So it means the cells checking and following the changes in DH ratio, and when it is increasing, that turn on hundreds of genes. And so it means by applying deuterium depleted water, we are able to inhibit hundreds of kinase genes. So we don't need to targeting one by one maybe 1,000 kinase genes. And earlier we published a paper when we treated the mice with a carcinogen agent that within 24 hours increased the expression of these genes. But when we kept the mice on a deuterium depleted water, we inhibited the increase of the expression of these genes having role in cancer growth. So the key issue, what you mentioned, by changing the DH ratio either with food or by consuming deuterium depleted water, we can control the expression of the hundreds of genes. And the probability whether the cells can start the cell growth depends on whether temporary increase the DH ratio, turn on maybe thousands of gene expression, and then in the next 18 hours, these express genes and proteins will manage it the whole cycle of the cell division.
Yeah.
This is how it works.
Yeah, that's really fascinating. It seems like there's this sort of constant flux based on the foods and the water that we're consuming with the DH ratio in the body. How is deuterium leaving the body? If there are a few selective mechanisms, you noted that the ATPase, the FO head, is very selective with taking hydrogen rather than deuterium. There's the sodium proton pump that is very selective as well. How is deuterium leaving the cells and ultimately leaving the body? I can only assume it's through the normal, urine, feces, and sweat it's coming out. Have you looked into how it's coming out and what might be responsible for getting that deuterium out of the cell and into the waste?
Only the chemical parts of this process, the amino groups collecting the deuterium. So and this is due to the isotopic effect, the carboxyl group will not keep the deuterium, but the amino group is collecting. And when we, for example, when we replace the water of the laying hens and we collected the eggs, and then from the eggs we separate the white and the yolk,
yep,
and we check the D level of the yolk and white, it was clear that in the protein the D level was higher at the day 0, which means the amino groups are collecting the deuterium. So the discrimination and the distribution between if I say carbohydrate, lipids, or proteins, the proteins has a higher DH ratio because the amino groups.
right,
and that way the urea and the nitrogen would be the way which chemically can remove and bring out the deuterium from the body. We never so we followed the D level of the urine and the saliva and the exhaled breath, and it was clear that not the same.
Yeah,
so
right.
it depends on what is the source of the body fluid.
Yeah. Have you looked into sweat? Is sweat selectively getting out more deuterium, or?
Not yet, but I read a paper who tested in the sauna whether
that's what I was thinking.
the patients consume this heavy water and then what is the distribution. And it was proved that, again, not was the same. And so there is some disbalance or something which finally, so we cannot say someone drink a glass of water or a half glass of heavy water, everywhere will be the same, the D level, so within the body. And for example, maybe the brain is which can, where the D concentration is increasing when someone drinking some heavy water. So the latest and the organ is the brain where the D level will increase.
Interesting. Yeah, because I mean, there's a lot of research coming out of Finland with the benefits of frequent sauna use, and I thought maybe there's an effect with all of the sweat that they're producing. Maybe there's more deuterium coming out in the sweat than would normally be present in the body. Maybe the sweat selectively excretes deuterium. But yeah, just a thought.
I guess the sauna is extremely good. Has the higher temperature, the metabolism is speeding up. Okay, one day we will check it out.
Yeah, all right, that would be great. I wanted to jump back to something you were speaking about before, how closer to the equator, the deuterium content is higher, and the further away from the equator you get, the surface water tends to be lower in deuterium. Clearly, we have human populations at various latitudes. And I was wondering if you had found any possible mechanism that would help people survive and thrive living closer to the equator, where they are consuming probably more carbohydrate, as there's more carbohydrate available at the equator, and drinking water that has more deuterium in it. You know, is there something in their environment that's helping them to balance the DH ratio differently than someone living in, you know, northern Norway, where the deuterium, their exposure to deuterium is much lower?
I guess that each population is adapted to the area where they live, the food they eat, and the water they drink. The big difference is today that we can eat banana, orange, which coming from the area close to the equator area, it means that a higher deconcentration. Or we can move from Norway to Caribbean island or whatever to the close to the equator area, which is a big shift in the deconcentration. And there are people who say that everybody should eat the food which is on the
local and seasonal,
getting from the field around them, and drinking the same water. So that way we again make a big disturbance in the metabolism and in the food and all the thing. Again, it's very important what type of food we give to the animals, what we are eating, what we eat. That way we can modify again the deconcentration of the final product, what we eat. I don't believe that on a short term that can cause problem. Someone goes to the holiday to Cyprus from Norway, that will not cause problem. But for example, in the Near East, the diabetes number is increasing. It's tremendous amount of diabetic people live now. No movement, no physical activity, too much carbohydrate, and that way I guess that should be one cause that the metabolic disease is so obvious, and almost 50% of our population is getting diabetic.
Yeah. Do you think there's any possible relationship between the intensity of UV closer to the equator perhaps playing a role in regulating deuterium dynamics in the body? I mean, there's more deuterium at the equator, there's more UV. Perhaps they work together to balance out and make sure things stay in the right level.
Yeah, that's we just took part in a conference, and Professor Pollack showed his interview, and he was very convincing with this easy water and the importance of the light for the body, for the skin, and for the humans. And what he mentioned that, so I was convinced with his presentation that so the water is not just moving, must be that structure, the hexagonal structure. And even after my presentation, I was asked that for the easy water, it's very advantageous if no deuterium, so no HDO out of within that, because even when there is a very complex system, this easy zone, all the atoms moving, harmonizing with each other. But when I replace one hydrogen with deuterium, that will disrupt this whole structure, because so different the vibrational frequency of the OD bond. So I guess the light is important, the UV is important, and that way. And I suppose now that if there is a deuterium within the easy zone, that will eliminate those water molecules molecule which has a deuterium, because doesn't fit into the system.
Yeah, that's fascinating. I suppose that's why the mitochondria is so selective with making its metabolic water, that it excludes deuterium. It seems as though
I
that
guess so.
the mitochondria are precisely where that EZ needs to be. It's cool that you bring Jerry Pollack up without me asking. I was going to ask about him and whether you're going to supply some water for him to use in some of his experiments.
Yeah, that was my idea also, to see whether using DDW, this easy zone is wider, build up much faster. Yeah, that should be an experiment. Yeah.
Yeah. I think it would be fascinating to see. I think it would help further his work as well, and help us understand a little bit more about what's going on in the mitochondria. So deuterium seems to be intimately connected to cancer metabolism, but does elevated deuterium levels in the body cause cancer, or is that just something that helps make things worse when exposed to a carcinogen?
I don't believe that the deuterium causes cancer. I think that the probability how cells are able to lift up the DH ratio is different and depends on the deconcentration of the body. So if someone eat too much carbohydrate, eating 150 ppm water, and for example, there is some genetic alteration, which, for example, increase the receptor numbers on the cell surface, that makes the cells easier. Or with aging, the mitochondrial function is getting lower and lower. So when the cells are getting older, then the capacity of the mitochondria to producing deuterium depleted metabolic water again slightly can increase the DH ratio, and this can help the cells from the higher DH level lift up to the threshold level, the DH, which increase the probability that one of the cells really can trigger the cell division and enter to the S phase.
So I guess that's why you're in the protocols, it's quite important to continue that deuterium depleted water even after remission, just to make sure that none of those cells have the capacity to continue growing if there are minute amounts left over in the body.
Yeah, this is one reason. The other reason, that when a tumor was operated, and everybody can be happy that, okay, it was removed perfectly, but when the tumor was diagnosed, the tumor were growing for 4 or 5 years in the patients. And from the original tumor, cells has removed and circulating in the body. And when the tumor is removed by operation, patients receiving the chemotherapy, radiotherapy, just to be sure that no tumor, but no one can guarantee. And we have to fight against those cells which are on the undetectable level. When someone making a cure in remission for 4 months, then we shocking these small group of cells and hopefully kill them. But again, we are not sure we killed all of them. So we suggest after 4 or 6 months break, start a new cure again. And hopefully slowly after 4 or 5 years, we really can eliminate all the potential cancer cells, which maybe cause again a new tumor.
And the lower deuterium content that you're trying to achieve with this therapy, like you said before, is a kind of stress on the cells that healthy cells seem to be able to handle better than cancer cells, which is why it's kind of like a selective treatment, which is sort of the holy grail in cancer. You want to be able to kill the bad cells while leaving the good ones alone. With that in mind, what is the sort of standard treatment protocol that you go through to make sure that stress stays continual, rather than going, directly to the lowest amount of deuterium depleted water, which isn't a good idea at all. You want to gradually lower the deuterium content of the body.
You're right. Yeah, thank you again for your good question. So for a cancer patients in remission, we recommend to consume prevent 105 ppm, 85 ppm, 2 months, 105, and then continue with 85. That should be good enough to reduce the incidence or the chance to relapse, and repeat it for the next 4 or 5 years. At the beginning, the break should be 4 months, and later 6 months, then every year repeating that 4 months treatment. Those who had a cancer and no complete remission, they should go 105, 85, 60, 20, 45, 25. The reason we say that, for example, someone immediately started 25, it means that after 2 months, they reach the equilibrium and will not be any changes in the deconcentration.
Yeah.
So all the cells which tolerated and survived the first 2 months, they are happy with the 25 ppm. And this is a big challenge. And this is, I try to write in my book, different protocols for different type of cancer and for different combination with the existing conventional therapy. And yes, we need lots of work. We need lots of new study to find the best dosage, depending on the cancer staging and all the things. So what is important message would be not the lowest, the best. And we believe that this gradual decrease, keeping the decrease on a long time, but still reaching the threshold and generating the radicals, which finally trigger the apoptosis. This is a big game.
Yeah.
And we have to learn it. And I hope one day I can educate the oncology society how to integrate deuterium depletion to the therapy.
Yeah, I was wondering, how has your work been received by oncologists? I mean, surely by now you've got people who are sort of taking your work to their oncologist and saying, can we incorporate this into our standard treatment? Are you getting any pushback from oncologists?
Yes. Yes. Yeah. It's not the same as it was 30 years ago. So I recognize. So this is we are changing the paradigm, and for a scientist who changing the paradigm have to be very, very cautious, waiting, because it is not obvious that they immediately pick up and accept it. So there are oncologists who knows it, but our aim is to have a registered cancer drug. So I don't believe that the way should be that people can buy it in the supermarket, deuterium depleted water, and they will consume it. We need a drug registered by FDA, EMA, and that should be part of the therapy. And on the other hand, we hope that we need a so-called functional food product, which would be lower than the natural water, but not in the range of the drug. And that people should consume it just to reduce the incidence of cancer, just to reduce the metabolic disease, or to feel better, because we tested with athletes drinking 105 ppm will trigger and have the mitochondria. So the lactic acid appeared later if someone consuming the 105 ppm. So this is a very long time. So yeah, it's 33 years. I started my research at the Institute of Oncology. We are getting close to the critical mass. We're publishing lots. I am invited to podcasts like you. Thank you. and for conferences. And one day we are waiting for investors. We are applying money to the European Union to support the clinical trials. And I hope we are getting closer and closer, and one day that will be accepted.
Mm.
You know the story with the Australian MDs with the Helicobacter pylori?
Yes, I do. I do.
Yeah, it took 12 years. They were MDs. They had a clear evidence that the connection between the ulcer and Helicobacter, and even it took 12 years.
No, even smoking. I mean, that took, I think it was 7,000 publications or something for it to finally change. So the progress is slow, but it is happening, isn't it?
I guess so.
Is this sort of treatment program that you outline in the book, is this something that you think people can do themselves if their oncologist is not on board with it?
Yeah, so that should be some kind of guide to do. And of course, there are lots of other aspects I try to combine. So if someone calls us, I try to give a very personalized advice, how to do, when to do, what to do. But in general, those protocols can be good. And even so, if we make the two groups receiving only conventional therapy and someone conventional plus anyhow drinking DDW, the result is much better than just receiving only conventional therapy.
Yeah. So it's something that you could implement yourself as well. It's going to help. It's just the degree how much it's going to help, I suppose.
Yeah.
Yeah.
So of course we have to use out the capacity which is in the DDW.
Yeah.
And the question how much people can use out based on the book, based on the knowledge, or maybe combining and discussing with the oncologist, because that would be the best. If they can discuss it and figure out the best way of the treatment.
Are there any sort of publications that you would think might be, if they're open access, or even if they're not, publications that might be good to take to oncologists and to introduce them to the research that you've been doing for the last 30 years, if they hadn't heard of it?
Our last paper was about the glioblastoma.
Yeah,
I don't know whether you have read it. So
I haven't read that one.
We evaluated 55 patients having glioblastoma multiforme, and again, how important the discussion. So we checked the median survival time for the whole population. It was 30 months, which means we can double the median survival time. But when we separated the people receiving the DW at the same time by receiving the radiotherapy, and those who already got the radiotherapy and after a couple of months started, then combining the DDW with the radiotherapy, the median survival time was 47 months.
Yeah, right.
And so it's very important when the DDW can join to the therapy and which stage, and so it's very important. So this is our last paper. Now we are going to publish case studies, 3 different cases. It is at the paper for waiting for publication. And one of the case study is a young girl with an astrocytoma 1 in the spine. No any therapy for that. No one. And she consumed DW. She was at age 11 when they started, and then they graduated. She finished the university, and she lived for maybe 18 or 20 years. And unfortunately, she had an embolia. After 10 years, she finished the DW, but it was clear that alone the DW consumption without any chemotherapy, any radiotherapy helped her. And the size of the tumor regressed, and she lived a normal life. So that hopefully will be published, will be accepted. in that paper, and there will be 2 other case study.
Yeah, that's amazing work. There's so many of these different case studies that you wrote about. Even in the first book that you published, I think in 2000, there's these case studies as well. But one thing that has stayed the same between the first and the second book that you've done is that there are some cancers that are a little bit more tricky. Melanoma and glioblastoma being 2 of the main ones. That's not to say that deuterium depleted water doesn't help. It's just a little bit more of a tricky situation. Is that correct?
Yeah, you are right again. So, for example, we don't like the pancreatic cancer.
Yeah,
the pancreatic cancer extremely aggressive, and we are just working on preparing a new paper. We are evaluating over 90 pancreatic cancer patients. The point is that I cannot be satisfied that we double the median survival time of the pancreatic cancer patients. To some extent, it's a great result, but when I started, the aim is not to extend the lifetime. The aim is to cure the people. And of course, we need lots of research. We need lots of scientists and oncologists to join to that type of work and to figure out how can we cure pancreatic cancer patients. And then we can say, okay, we did what we had to do.
Yeah, absolutely. It's a really, really huge problem that we're facing at the moment, which I think is why your ideas and your research over the last few decades has been so important. I wish more people knew about your work, which is why I'm trying to get more people aware of what's going on, mostly in Hungary, I think, but it's starting to branch out into the States as well. I've noticed. So, have you noticed it picking up? Is there a bit more of a buzz around deuterium depletion at the moment?
No, I guess we are getting in a much better position. So the book is very helpful, and the 30 years is finally 30 years.
Yeah.
And hopefully, and this is the best, just like you, lots of people is going to support that and help that process, because the interest is to solve that type of challenge. So the society is facing with lots of other real big problems, and the cancer is one of them. The prices are increasing, the incidence is increasing, and no real solution. So we must solve that problem. And thanks to everybody who helped me and helped that more people should get information about this research program and all the data and the results that we already have. So thank you again for you to do that.
No, thank you. I'm happy to play my part in sort of spreading the word. I guess it would be good to mention that the product that you have been working on is called Vitera, Vitera 25.
So Vitera is the cancer drug for veterinary use. And the Preventa is a drinking water for human consumption.
Yeah, so Preventa 25, it is a result of a very, very arduous process, if I understand correctly, which is why the water is rather expensive. And I think you've been working on lowering the price of that and making it more available very, very hard over the last few decades. So hopefully in time, with some grant money and some support from hopefully your government over there, the price can come down a little bit. But no, we have a distributor in Australia here. It is available if people want to check it out. I'll make sure I leave links to all of your work, your lab, and links to the books as well, so that people can get their hands on them and maybe give them to their doctors and whatnot as well.
Thank you very much. Thank you.
Thank you so much for listening, everyone. I really hope you enjoyed this one. If you're interested in reading Gábor's new book or watching some of his presentations on YouTube, or even finding some deuterium depleted water for yourself or someone you know, I've left all the relevant links in the description of this episode. Feel free to share his work with doctors and oncologists, as this work needs to be more widely acknowledged. If you'd like to support this podcast, you can subscribe so you get notified whenever I release a new episode. I'd also like to encourage you to leave a 5-star review or give a thumbs up if you like this episode. This is a simple, no-cost way to support my work and help me reach more listeners. Please feel free to leave comments on my YouTube channel, as I do try to read through as many as I can. I've also put links to all of my social media platforms in the episode notes if you'd like updates about the podcast, information about health, or if you just want to reach out to me in general. Thanks for listening, everyone. I really hope you enjoyed it. Take care.